Welcome back to another episode of Advanced Gross Human Anatomy. Today is our fifth dissection, and we will be looking at, we left off kind of looking at the spermatic cord as it goes through the inguinal canal. Today, we'll be following that spermatic cord into the scrotum and also doing some dissection of the abdominal wall a bit further. So, as a review here, we looked last time at the spermatic cord exiting the inguinal canal through the superficial inguinal ring, right about here, as it continues down into the scrotum. The testes, though, as we look a little developmentally, they form, here's kind of a progression of the development of the testes within the male. They form up in the abdominal cavity and by the early forming kidneys. You can kind of see where they are up here, where they would be. Initially, they're much larger in proportion, the testes are, to the size of the fetus. As the male fetus continues to grow, you can see they become proportionately smaller to the rest of the organs. They also begin to descend. You can see how high they are up here. They start to kind of come lower within the abdominal cavity as the cavity itself grows. So partly it's that the surrounding structures and the cavity itself are growing, and they kind of draw them down. But there's also this ligament, this gubernaculum testes. It's a funny word, gubernaculum testes. It's this ligament that connects the testes to the abdominal cavity, within the abdominal cavity, to the labial scrotal swelling, which is kind of on the bottom part of the perineum of the male. And when those two labial scrotal swellings come together, when they fuse together, they form the scrotum. And in that last month or so, you can see it coming down here, in that last month or so of pregnancy, the gubernaculum testes actually shorten and pull the testes down through the inguinal canal down into the scrotum on each side. You can see that gubernaculum testes here pulling down into the scrotum. So developmentally, they actually start up in the abdominal cavity before they get pulled down. If we look at the scrotum again here, we remember from last time, this is a pouch found on the outside of the abdominal cavity. Remember that optimal temperature for spermatogenesis is about 2 to 3 degrees Fahrenheit below or cooler than body temperature. And so the testes will change, the scrotum will either bring the testes closer to the abdominal cavity or allow them to drop further from the body wall, depending on the temperature of the testes themselves. A couple of muscles that we talked about before, the cremaster muscle, which is found within the spermatic cord. We'll touch on that a little bit more today. And then the dartos muscle, which is in the wall of the scrotum itself. Let's see, this is probably there's a better picture in a little bit here, but the dartos muscle within the scrotum itself, these two together will contract, and then when the testes are chilled, or when they're cooler than they would like to be, and they will draw the testes closer to the abdominal wall. Conversely, when the temperature is warm, when they're a little too warm for optimal spermatogenesis, the cremaster muscle within the spermatic cord on each side and the dartos muscle within the wall of the scrotum will relax and allow the testes to drop further from the abdominal cavity, from the abdominal wall, really. If we look at these layers, you can kind of see on this, there's distinct layers of tissue, 3 layers specifically of tissue around that spermatic cord, surrounding the spermatic cord. And this is formed as the gubernaculum testes pulls the testes down from the abdominal cavity. It has to go through these abdominal layers here as it pulls through the inguinal canal. And it picks up these layers. There's an internal spermatic fascia, there's an external spermatic fascia, and then outside of that is a cremasteric fascia. So initially you can see this internal spermatic fascia on the inside here. comes from the transversalis fascia. Remember that we talked last time about this layer on the inside of the muscular wall of the abdominal cavity. It's that transversalis fascia. So as it pulls down, that's the innermost layer that it's gathering. The external spermatic fascia is where it comes from the external oblique aponeurosis. As it goes, as the gubernaculum testis pulls down through, it will grab a hold. It'll pierce through that external oblique aponeurosis, where it covers the rectus abdominis, and that will kind of pull down. That's the external spermatic fascia, and then the cremasteric fascia comes from when the testes goes through the internal oblique muscle, right, as it enters into that deep inguinal ring here. And those three layers will cover the spermatic cord, from the innermost, which is the internal, and to the outermost, which is the cremasteric fascia. Now, that cremasteric fascia, here's just a different shot of these layers, these layered systems here. But the cremasteric fascia, this outermost layer, makes that cremaster muscle. So that's one of those mechanisms that will help pull the testes closer to the abdominal wall as the temperature changes. You can also see this plexus here, these blue lines, the vasculature here. It's called the pampiniform plexus. It's on each side, obviously. The cremaster muscle is just not dissected on this side, so you can't see it. But the testes aren't drained by a single vein, but rather a whole plexus of veins that drain the blood from the testes. So that's that pampiniform plexus. Also, within the spermatic cord, you'll have the testicular artery coming down and supplying blood to the testicle itself. You'll also have the vas deferens in there. Vas deferens is a muscular tube that has its own blood supply, interestingly. It has an artery that travels right alongside of it called the deferential artery. The vas deferens and the deferential artery follow along with that to give it its own blood supply. And this can be helpful, I'm told this can be helpful in identifying the vas deferens when you're doing a vasectomy for the sterilization process in the male. When you would cut the vas deferens, one of the things you can use to identify where that vessel is that it has that deferential artery. Now, I certainly I'd be a little nervous to do that for the first few times. Cutting the vas deferens, to me, when you dissect this on the cadaver, I have a hard time sometimes differentiating the testicular artery from the vas deferens. Those two vessels look fairly similar to me. Maybe in live tissue, this is different, but I imagine that's one of the features that allows the clinician to identify the vas deferens and not cut the testicular artery. Boy, you wouldn't want to do that in the process. A couple of things here with pathology. Because of this pampiniform plexus here, a couple of things that can happen that will cause necrosis of the testes. The pampiniform plexus is one area. If there's a varicosity, like a varicose vein, part of that pampiniform plexus can grow and put pressure on the testicular artery and block blood flow to the testes. That will cause necrosis of the testes. Another way is through a hydrocele. As the testes descend through the spermatic cord, there's a space formed. You can kind of see this space formed up here called the processus vaginalis. If this remains patent, and fluid collects in there, that can also put pressure on the testicular artery and limit the blood flow to the testes. So a couple of interesting ways that this could lead to a pathology if there's hydrocele or varicosities. Let's see. The testes, you can see here's a cadaveric view of the testes. It's covered by a white coat. In Latin, it's called tunica albuginea. Tunica means coat. Abugenia means white. The tunica albuginea. Both testes and ovaries, male and female gonadal organs, are covered by that white coat, that tunica albuginea. You can see this pouch on the side of it here. This is the epididymis, found on the side of the testes. that contains a coiled network of tubules, which when stretched out is actually about 25 feet long. There's a lot of distance there to travel for those sperm cells. The sperm cells will, within that epididymis, will gain their maturation, but they'll also be stored until ejaculation into the vas deferens, where then it will leave the scrotum and then come up. But we'll touch on that maybe in this slide here. So looking at the testy itself from this drawing, you can see the tunica albigenia on the outside here, the epididymis on this pouch on the outside of the testy itself, all within the scrotum. You can see these ducts, these seminiferous tubules, where the sperm is produced. So spermatogenesis starts in the testy itself. Sperm cells are morphologically mature when they leave the testy and go into the epididymis, but they aren't quite fertile yet. They're not mature enough to fertilize an egg. Somehow, they gain their maturity as they travel through the epididymis before they would be ejaculated out. So within the epididymis is actually the site where sperm cells gain their maturity. But they're stored within the epididymis until the ejaculation into the female reproductive tract. That's where they will be stored through here. Then, once they are pushed out of the epididymis here during the ejaculation process, they go through the vas deferens. Here you can see the deferential artery attaching to it here. They will go through the vas deferens. That goes up through the deep inguinal ring of the spermatocord, into the pelvic cavity, through the deep inguinal ring, all the way behind the bladder to the back bottom portion of the bladder, into the ejaculatory duct of the prostate, and then into the urethra of the penis, so that it can be expelled out of the body. It's a long ways. You think some people would think that, oh, it's right next to the penis. I bet that's just a quick little route here. It's actually not. Actually, it has to go over the bladder, through the inguinal ring, into the abdominal cavity, through the inguinal canal, into the abdominal cavity, through the deep inguinal ring, around the bladder, bottom part of the bladder, into the prostate, into the urethra, and then finally exiting the body. So quite a long ways to travel for those sperm cells. Let's talk about lymphatic drainage of the scrotum now. This gets interesting when we talk about pathologies as well. For further drainage of the vasculature, but also in case of infection, you've got a couple of nodes here. The superficial inguinal nodes, which are kind of these nodes in the inguinal region. These are for draining lymphatics from the penis, from the genitals, and the scrotum around here, but not the testicles. Okay, so more for the penis and the scrotum, but they're going to be on the sides here in those superficial inguinal canals. There are other nodes receiving lymph drainage from the testes that are higher in the abdominal cavity. You can see all these up here that are near the lumbar aorta. And this is an important consideration for testicular cancer. So the testes don't drain into the inguinal nodes. They do the superficial inguinal nodes. They'll drain up into the abdominal cavity, into some lateral lumbar nodes and some pre-aortic nodes up into here. And this gets tricky if you've got testicular cancer that's traveling up into the abdominal cavity. Then those cells, those cancer cells, can travel up, and go way up, even into joining into the thoracic nodes around the aorta and vertebral column. So, if there's issue with finding that where the testicular cancer has stopped its metastasis process, you want to catch that quite early. So that it doesn't travel into those nodes, into the thoracic, or even into the abdominal or even the thoracic cavities. Let's switch gears a little now and talk about the abdominal cavity. So the abdominal cavity, we've already dissected the anterior abdominal wall. You've gone through the musculature, the skin, and the musculature of the anterior abdominal wall. The abdominal cavity is continuous with the pelvic cavity below. It's the superior portion. Sometimes you'll call this the abdominal pelvic cavity, but it's the abdomen and the pelvis, anything down below that in there. There's a peritoneum, which is a membrane that surrounds all of the abdominal cavity, kind of separating it from the muscular wall here into the abdominal cavity as a peritoneum. It's a serous membrane, and a serous membrane secretes a lubricating fluid. It's very watery, very thin, it's very slippery. It allows the abdominal contents to be able to slide on each other during movement. I'm not a hunter, but if you're a hunter, maybe you've seen this when you've field dressed a deer or something. When you cut the abdominal contents out, they just kind of slide out on each other. They all just kind of gather in. They're contained within that peritoneum that has that serous membrane that makes it really quite slippery. So when you breathe and when you jump and when you move, that allows these organs to slide and glide on each other and not get stuck to one another. Different than a mucous membrane that creates mucus. This is that serous membrane that creates that lubricating fluid. And then behind the space here, there's also the space that's retroperitoneal. So there's the peritoneum, which is where all the abdominal contents are, minus a couple of areas, which we'll talk about. And then behind that is an area, this space called the retroperitoneal space, behind the abdominal cavity. Here in this picture, you can see these membranes holding the intestines to the back of the abdominal wall here. These are called mesenteries. So they will hang on to portions of the intestines. If you're wondering, this looks a little funny. If you see all these holes here, all these vessels, these tubes, think of this like if you wound up a garden hose and you were to cut it in half as it was wound up in nicely on the yard. If you took a saw and you cut that right in half, you'd have all these tubes looking at you, but it's just one tube, right? But it's just cut in cross section. So here you can see a sagittal section. That's what all these tubes are here. But sections of those intestines, these mesenteries, hold it to the posterior abdominal wall. There's also on this slide, you can see a couple of things here. Different types of membranes that connect organs called omentum. The omentum, there's a lesser omentum here connecting the liver to the stomach. And there's a greater omentum coming from the stomach that comes all the way down, drapes over the anterior abdominal cavity, and then wraps back around to attach to the transverse colon. That's called the greater omentum. There's also various peritoneal ligaments that kind of connect connect tissue membranes. These are connecting various structures that hold them in place within there. But the things, the two big things: mesentery, holding the intestine to the posterior abdominal wall, and then the lesser omentum. Remember, omentums connect organs, and mesentery connects organs to the posterior wall. But the lesser omentum up on top here, and then the greater omentum draping over as it comes back to attach to the transverse colon. Here you can see the diaphragm on top, separating the thoracic cavity from the abdominal cavity. Let's see here. The diaphragm would sit on top here of the liver on the right side, and the stomach and spleen on the left side. It's not shown on here, but that's kind of where that is. That cross section just kind of showed the liver. But here you can see how it would cover the spleen and the stomach on the right side, on the left side. Right side, liver, left side, stomach and spleen. Here you can see just a picture of some of the organs that we'll talk about now. Under the diaphragm on the right is the liver. That's the big one here. Let's go to a different shot here and see the liver by itself. It's got a couple of lobes. You can see on the front side there's a left lobe, which is a little smaller, a right lobe, which is much bigger here. Underneath the diaphragm. And if you flip this over, here you can see how, imagine this just kind of flipped on its end. It's just going to flip over this way. So here's the right lobe, here's the left lobe. Again, but here's a couple more lobes here. You can see a quadrangular lobe, kind of a square-shaped lobe, more inferiorly. That's this portion here. It's kind of by the gallbladder. It's called the quadrate lobe. Think of that as quadrangular. It's kind of a square, quadrate lobe. And then more superiorly, by the inferior vena cava here, toward the back, you can see the caudate lobe. Maybe that's caudate towards the back. I'm not sure if that helps you. I'm not going to go too deep into the details of the four lobes. All of the lobes have the identical function. So this would be helpful to know left, right, quadrate, and caudate lobes of the liver if you're going to be performing some type of procedure, or if you're trying to investigate particularities within a lobe itself. That might be helpful. So there is some value to learning those, but for our perspective, functionally they all act identically. On the top here, on the anterior portion, you can see again this falciform ligament. We talked about that in a previous dissection, kind of separating the left and the right lobes here. This attaches the liver to the anterior abdominal wall. If you continue down from the falciform ligament, you'll see the ligamentum teres. Again, we talked about that prior in a previous dissection. This is the inferior aspect of the falciform ligament. In the womb, remember, this is the blood supply from the placenta to the fetus. And after birth, it kind of closes down, that lumen closes down and becomes this ligamentum teres. Remember, teres means rope, so it's like a rope-like ligament coming into there. Okay. I'd ask if there's questions, but that might not be helpful. I'll just keep moving on here. Let's talk about the spleen. Spleen's this organ. It's kind of this gray organ on the picture here. Here you can see the liver underneath the stomach in front here, more on the left side, and then the spleen. The spleen is enough. It's about the size to kind of fill your hand. If you pass your hand under the diaphragm on the left side and go behind the stomach, you'll basically be holding the spleen. Otherwise, it's kind of hidden and out of sight here, but it's kind of behind that area. It functions as a blood depot. It kind of stores red blood cells. It's also a lymphatic organ. It does have some white blood cells as well. It's dangerous, though, because of its storage of all the red blood cells. If you do sustain a blow to the left side of your back, kind of a kidney punch blow, if the spleen ruptures, you could lose a lot of blood internally very quickly. So that could be quite dangerous there. If we take a look now at the stomach, here's the stomach. I'll get a better view of that. I'll just isolate it here for you. So the stomach, as the esophagus comes down, people think, well, here, let's go back to this shot here. People think, oh, if I were to say, where's your stomach? A lot of people would say it's kind of right in the middle, right below their diaphragm, right above their belly button. And it's actually more to the body of it's more to the left of midline. You can see here. But the stomach lies mostly on the left side of midline underneath the diaphragm. It's got a few regions here. You can see where the esophagus is coming down. This is the cardiac portion of the stomach, where the esophagus enters. In this area, mucus will be formed to protect the stomach lining from the acidic environment. So that's going to help. Some of these gastric juices are formed kind of in through here. You can see this portion on top, this bulge on top, more to the left of midline from the cardiac region, is called the fundus. It's a bulge on top. This collects air from as you swallow, or if you drink carbonated beverages, possibly, or you can get kind of some accumulation of air in through here. So you can see it'd be kind of like a balloon, kind of gathering up this air into the top here. And then as the diaphragm pushes down on this, either when you're talking or laughing or breathing, it's going to push down, and so that air that got trapped up in the fundus is going to come out through that cardiac region, back up the esophagus, and that's where we get a burp or a belch. Comes from that air trapped in the fundus. The body seems pretty natural that we'd call the major portion of this the body, just the body of the stomach. And then at the distal end, as it comes down here, as it enters into the duodenum, the proximal portion of the small intestine, there's this pyloric region. Here's a cross section of that kind of a coronal view of that pyloric region. This distal end that narrows. You can see it's quite narrow at the end. So as the stomach, as the body comes down, it kind of funnels in, and then the contents of the stomach will enter into the small intestine through that pyloric sphincter here. That sphincter helps hold food in the stomach for it to be churned up by the musculature of the stomach prior to being passed on to the small intestine. This allows it to go much slower, allowing us really to not be requiring to eat continuously through the day. So it kind of holds the stomach contents so that it can pass through much slower. We talk about let's see, the musculature of the abdomen, of the stomach. We'll just keep it on this picture for a second. The musculature is interesting. Characteristic of the rest of the GI, you've got two distinct layers. There's an inner circular layer, and then there's a outer longitudinal layer. And those two layers are important for peristalsis. And they help kind of push the food down. So that circular layer, that inner circular layer is going to kind of, let's talk about the outer longitudinal layer first. The outer longitudinal layer is going to kind of make a bulge within that the GI tract, and then the inner circular layer is going to kind of push that material, that bolus of food through the bulge, and that's how we get that peristalsis. But the stomach is interesting because it's actually, the process of mechanical digestion that was started in the mouth actually continues down into the stomach. So the food is actually mechanically being digested as well as chemically in the stomach, but it's all mechanically being digested in the stomach as well. And there's a third layer, an innermost layer, which is an oblique layer. So here you've got this outer longitudinal, this middle circular layer, and then this innermost oblique layer coming on the inside. So those three layers contract at these angles to each other to continue that mechanical digestion in the stomach. And you just need a little bit of air and food in here for that to happen. So if you're getting some mechanical digestion without a lot of contents in here, you'll get some of that growling, or your stomach will make some noise as the air starts to move around there with a little bit of food. It doesn't take much for that to start. What I'd like you to do today in the dissection is here's a cross-section, kind of a coronal view of the stomach. What I'd like you to do in the dissection today is open up the stomach so you can see the inside of it. And so what you'll do is you'll kind of cut from the body. You can kind of up toward the fundus if you'd like, but up toward in the body, cutting down through kind of the midline of the anterior stomach, and cut right through the pyloric region, in kind of starting into the duodenum. We're going to also cut today from the duodenum down. So, but for right now, if you just kind of cut into that pyloric region so you can see, and what you'll notice are these rugae, these folds, these gastric folds within the stomach, these wrinkles in the interior stomach wall. And that allows the stomach to expand. So when you're eating maybe a big meal, it allows the stomach to grow, and so it's going to be able to expand as the contents increase within the volume of the stomach. Let's look at, let's see here. Another shot of this. A couple different terms here. We talked about the lesser omentum on that one picture. Here's another shot of the lesser omentum coming from the liver, coming down to the lesser curvature of the stomach. The lesser curvature is a smaller one up on top. There's also called the greater curvature on the bottom, which is where the greater omentum is coming into. So lesser omentum, greater omentum, corresponding with the lesser curvature and the greater curvature of the stomach. But the lesser omentum is also called the hepatogastric ligament. It's this connection between these two organs. And that from the liver, there's another one here similar to it, from the liver to the duodenum right here, so not to that lesser curvature of the stomach, but to here is called the hepatoduodenal ligament right here, and that contains the bile duct from the liver from the gallbladder rather, contains the portal vein, which brings up all the blood from the intestines to the liver, coming in from the portal vein. It also contains a branch within this hepatoduodenal ligament. There's a branch of the hepatic artery, which brings oxygenated blood to the liver. So all of that's going through here. So the lesser omentum, or the the hepatogastric ligament, and then you've got the hepatoduodenal ligament next to that here, coming from the liver to the duodenum. Let's see here. In a future dissection, just to talk briefly about this, in a future dissection, we'll be kind of tearing through the lesser omentum to expose part of the abdominal aorta, specifically the celiac trunk, will be deep to here. So we'll be able to look at that a little bit later on. Here you can see the greater omentum as well. Again, you can see it contains a lot of fat, and you'll notice this as well. It arises from the greater curvature of the stomach again, comes down over the anterior abdominal cavity, loops back up and around and attaches to the transverse colon. Right here you can see the transverse colon. It's going to loop down like a curtain, kind of coming up and around. And this helps wall off the stomach and helps contain infections from spreading within that abdominal contents, within the abdominal area. The abdominal organs can also be a site of metastasis, and so this is often removed in surgeries that are trying to remove some of the cancer spreading cells through there, that greater omentum. Okay, well, let's keep rolling on here. We'll talk about the small intestine. The small intestine has three parts to it. The duodenum is the proximal portion, right off of the small intestine, just distal to the pyloric sphincter is the duodenum, kind of encircles the pancreas here. And then you have the jejunum, which is the proximal, about 2/5 of the small intestine, and then you have the ileum, which is about the distal 3/5 of the ileum, and then that will come into the cecum, which is the first part of the large intestine. So we'll come back around there. We're going to cut a portion of the small intestine. Let's see, what I'd like you to do today, so just pick any kind of portion of the small intestine. Maybe look for an area that doesn't look like it has a lot of contents in it. That might be helpful. And what you want to tie off though between the cuts. So maybe 3-4 inches or so of space. Get some string, tie off the small intestine, kind of puncture through the mesentery on each side here, and just tie it off, so that we can constrict that, so that contents aren't coming down from the top or up from the bottom. We want to cut kind of a longitudinal section through, just cut through that small intestine, and we'll clean it out and be able to kind of notice some of the things that are inside of there. So the small intestine is a primary site for digesting food. And so if you're digesting food and absorbing nutrients, you want to increase the surface area of that space tremendously, and that happens in the small intestine. And it happens primarily, you want to, one way to gain distance or space or surface area for digestion is just the length of the small intestine. It's quite long. It's about roughly about 22 feet long. So just the length of it is the first way we can maximize our surface area. The next way are these circular folds, these plicae circularis. Plicae means folds, circularis means circular, so circular folds in Latin. Plicae circularis. They're not smooth. Like the wall of the stomach, you should be able to kind of contrast this to the wall of the stomach, which, let's go back here quickly. It's quite smooth. It has the rugae, those ridges, but it's very smooth in its appearance. Contrasting that to the small intestine has these plicae circularis. Now, on top of those circular folds are villi. Now, villi are like a microscopic shag carpet. It has increases the surface area significantly, and then they have columnar cells on top of the villi called microvilli. So even more surface area, exponentially more surface area on top of the villi, on top of the circular folds. So all of this is to significantly increase the surface area within the small intestine, so you can see for digestion. Let's see here, the duodenum. Let's go back to this. The duodenum, I believe it means 12 finger widths. It's like it's a unit of measurement, I believe that name comes from. But the duodenum is retroperitoneal. It's behind the peritoneum. Here you can see how it's going to, you can see the peritoneum, the edge of it here. As the pyloric sphincter enters the duodenum, it comes posteriorly in the abdominal cavity, so behind the peritoneum, and then it comes up and around, kind of circling the head of the pancreas. Here you can see. The pancreas is also retroperitoneal. Pancreas makes enzymes that help digest food that get released into the duodenum, but that's going to be what we're looking at here. Within the cells, let's see here, if I look at the duodenum from a kind of cross section, within the small intestine are cells called crypts of Lieberkühn that help influence the chemistry within the small intestine. Also, there are within the duodenum, there are duodenal glands that will help shift the pH from the acidic environment from the stomach. So when the stomach was very acidic, to break down some of those proteins. As the contents come into here, some of that acidic environment needs to be transferred into more of a basic environment, more of an alkaline. So these duodenal glands will help with that as well. The common bile duct, you can see coming down through here, is going to enter into the duodenum from the gallbladder and the liver. Bile will help start emulsifying fats and allow for absorption of those lipids into the small intestine. Carbohydrates start getting broken down pretty much immediately in the mouth with salivary amylase. They continue to break down in the stomach. They break down quite quickly, relatively speaking. Proteins start breaking down with the gastric juices, hydrochloric acid and such within the stomach. But fats don't start getting emulsified until they hit the duodenum here, and then that bile coming from the gallbladder, which is storing from the contents of the liver, will enter into the duodenum, and that's what starts breaking down, emulsifying the fats. Let's see, what else do I want to talk about here? So let's see. Yeah, so basically mechanical digestion has stopped now, right? So now we need the enzymes from the gallbladder, we need the enzymes from the pancreas. So mechanical digestion from the mouth and the stomach is now leading to just chemical digestion within the small intestine. So that's what's happening here. Now, in this dissection, I also want you to tie a string maybe around here on the duodenum, maybe about halfway down, maybe 6 or 8 inches down onto the duodenum. And then I want you to cut open from the pyloric sphincter. Just continue that cut that you made from the stomach down into that proximal portion of the duodenum. And what we're looking for here is to contrast the appearance, again, of the internal wall of the stomach, which is real smooth. And then here you should be able to see the villi of the duodenum. Kind of gives that velvety more appearance. I also want you to look for, when you open the duodenum here, to look for 2 bumps. Well, one in particular, one big bump here, larger bump called the major duodenal papillae, also called the ampulla of Vater, which is where the entrance of the common bile duct is coming in, joins with the mass of the pancreas, this pancreatic duct coming in, that will enter into the duodenum. Just above that, sometimes you don't need to look too hard for this, but sometimes you'll find a smaller bump, a minor duodenal papillae above that, kind of an entrance of a smaller pancreatic duct entering into the duodenum. But the major one here, the major duodenal papillae, the ampulla of Vater, is the kind of proper name for that. All right, that's the duodenum. So a couple of cuts there that we'll investigate. As we look down into the distal part of the small intestine, then from the duodenum starts the jejunum, and then going into the ileum. Now, there's not a clear line of demarcation. There's not a real good way to differentiate between the where the jejunum stops and where the ileum starts. But the lower you get in the GI tract, the slower the peristalsis gets. So it's getting slower and slower as it as it goes through the small intestine. So what you might see here in the proximal portion of the jejunum is going to be more red, a little larger in diameter. The ileum might be a little smaller in diameter, a little more yellow in appearance. Maybe this is a little more easily seen on live tissue. If you're doing a laparoscopic procedure, you might be able to kind of notice some of those nuances, some of the differences. But for our purposes, they might be kind of subtle. So you don't need to look too closely at those differences there. But let's move on to the large intestine now. As we look at the large intestine, think of it like a frame. So you can see the ileum coming into here. It will enter into the cecum, which is the proximal portion of the large intestine. You'll have an ascending, a transverse, a descending, and a sigmoid colon, and then the rectum, and then the anus. So that is your large intestine. But you can see how it makes a bit of a frame surrounding the abdominal contents. Now, if the small intestine's function was to absorb nutrients, the large intestine's function is to absorb water and electrolytes. So the nutrients have already been absorbed in the small intestine. Now it's the job of the large intestine to absorb that water. So it's a very slow movement of the material through the large intestine. It takes hours for this to happen, to get through from one place to another. If you're sick, if you've got an upset stomach, we like to say, oh, I've got stomach problems. Nobody likes to say I have diarrhea. I've just got some stomach stuff going on. That's kind of what we like to say, just for to save us the embarrassment. But we all know what we're talking about, right? If there's too rapid of movement of material through the large intestine, enough water won't get absorbed back into the body, and so you'll have diarrhea as a result. That the water content will continue to wash through and then exit out, and you'll have diarrhea. What's interesting is what takes seconds. If I go back to here, it's to get from your mouth down into the esophagus, down into the stomach. It just takes seconds. If you swallow something, you can sometimes even feel if it's cold or hot. You'll feel it kind of coming down into your chest, and then you kind of lose that sensation as it hits your stomach. But that just takes a couple of seconds. What takes seconds to travel that maybe 8 or 10, 12 inches from your mouth to your stomach might take hours to travel that same distance as it comes through. So again, it's getting slower and slower and slower moving as it enters in more distally into the large intestine. Now, just for our purposes here, what I want to see here is that the difference between the colon and the large intestine. The colon has 4 parts: ascending, transverse, descending, and sigmoid. The colon's part of the large intestine. The large intestine is the cecum, the colon, ascending, transverse, descending, and sigmoid, and the rectum and the anus. So the sigmoid, cecum, rectum, and anus, and the colon all make up the large intestine. So just to get those straight, there's a valve here. As the ileum enters into the cecum, there's a valve, kind of a check valve to stop material from coming back into the small intestine once it enters into the cecum. This is helpful. Think of vomiting. If you're getting if you're really pulling from a deep place here, you're going to vomit things from the stomach and some of the small intestine, but not the large intestine material. Diarrhea is going to happen in the large intestine. Vomiting is going to happen from the stomach and the contents from the stomach and the small intestine, but that valve kind of separates those two areas there. Off of the cecum, you'll see an appendix, the vermiform appendix. Vermiform means worm. It looks very much like a worm. You'll be able to see that. We're going to examine the large intestine today, but in a future dissection, we'll look more closely at the vasculature that supplies the large intestine. But that's really all I wanted to see. You're just going to be able to kind of notice it today. Just see the structures, notice where they are. A couple more things on the terms here. The right colic flexure, the colic flexure here, where the ascending colon makes a right turn here and becomes the transverse colon. also called the hepatic flexure because it's by the liver. As the transverse colon makes a turn down again, turning right down into the descending colon, the transverse turning into the descending, that's the left colic flexure, also called the splenic flexure because it's by the spleen. So you can see that. The transverse colon, you can see here, has this mesentery, this transverse mesocolon that will help hold it to the posterior abdominal wall, and the sigmoid colon as well has this sigmoidal mesocolon. It doesn't have a mesentery, that's more for the small intestine, but the mesocolon here connecting the sigmoid colon and the transverse colon to the posterior abdominal wall. Remember when I said there was a couple of things that are retroperitoneal? Remember that the duodenum is retroperitoneal, so that comes out, but then it comes back. The small intestine is within the peritoneum, but then it comes in. The ascending colon and the descending colon are retroperitoneal again. So they're behind the peritoneum, and so the transverse colon and the sigmoid colon are within the peritoneal cavity. So you'll be able to kind of notice that, and the connections of that to the posterior abdominal wall, similar to the mesentery in the small intestine, are the sigmoidal mesocolon and the transverse mesocolon. All right. If we look at the last portion I want to talk about here is the rectum. Now, in the going back to this picture, we've transitioned from skeletal muscle within the esophagus down into smooth muscle. So skeletal muscle in the mouth and in the esophagus, we changed to smooth muscle into the stomach and then into the small intestine. Large intestine, still smooth muscle. But in the rectum now, we changed from smooth muscle back to skeletal muscle. So that's just a bit of a difference here. So you can see it's more skeletal muscle. Here it's broken up into little compartments here. You can see how the rectum, so here's the sigmoid colon, and then now it transitions to skeletal muscle, but it's broken up here into these sections, these compartments. Fermentation is occurring in the distal part of the large intestine. You can gas pains, I forgot to mention that here, gas pains as it comes down into the descending and sigmoid colon are often on the left side of the abdominal cavity. If you've got pain on the right lower quadrant, it might be an appendix issue. If you've got pain in the left lower quadrant, you might have gas pains. So that's kind of coming down into there. So as the materials the water is getting absorbed, fecal matter is forming through the large intestine. As it comes into the sigmoid colon and enters into the rectum, stretch receptors are found within the walls of the rectum, and they will stimulate the urge to go to the bathroom. Luckily, you also have alert signals going to the brain, so that we can contract these sphincters on the bottom here in the anus to help hold that material down. These sphincters will help hold the material so that we can find a more socially acceptable place to void our colon. But if you kind of contract these sphincters, if you don't answer that call of nature and find that bathroom quickly, you will fecal material will get pushed backwards, and pushed back into the sigmoid colon. Until more material starts pushing on the backside and the descending colon, and pushing more material, and you'll have a more urgent call to go to the bathroom. So that's where that's kind of happening and through here. Otherwise, if you're a baby and don't have control of these sphincters, and that communication is not happening in the nervous system as well, then you'll just fill your diaper. Hopefully it's a diaper. So you can see there. Let's see, at the end here, last thing we'll talk about, the end of the rectum in the anus are these anal glands, right at the end of the rectum here, as it enters into the anus. These anal glands have these pockets. They kind of form these sinuses between them, right? You can see kind of in through here, and that's where these glands will create a mucus that accumulates within those pockets, so that when fecal matter passes through, gets compressed in this portion and then gets lubricated by the contents of those glands, and allows it to pass through the anus more easily. So if you have issues with that, if you need some type of suppository or something that's going to help lubricate that area a little bit better than the glands are doing on their own, that's another option for that. There's ways to get around that, but that's what's happening within these anal glands. They do have that mucus does have a bit of an odor. Maybe you have an animal that has, like a dog, that has to have its anal glands released. Something's happening, kind of an infection or accumulation of that mucus within the anal glands here. Okay, the last thing I wanted to talk on here, last term here, these 10A coli. Remember we talked about those longitudinal muscles within the large intestine. These have kind of a segmented appearance. You can see how it kind of holds. It's kind of like it's bound here, like it's tied. You can see these longitudinal cuts. They give it a segmented appearance that has a name. They're called haustra. These segments formed by those longitudinal muscles, the 10A coli, 10A, T-E-N-I-A-E coli, C-O-L-I, within the large intestine. And then you've got these, you can see these fat stores here. You'll notice on the large intestine it's quite fatty. So fat stores are there. These are called the epiploic appendages, or appendices epiploica in Latin. The epiploic appendices for fat storage, right there. And that, I believe, is all that we'll cover today. I hope that was clear. Hope you learned something, and looking forward to seeing you in lab to look at this a little more closely. Thank you for your time.